What's the difference between prokaryotes and eukaryotes and how DNA synthesis is initiated, how it starts? Prokaryote versus eukaryote. Bacterial chromosomes, circular. Eukaryotic chromosomes are linear.
Isn't it like for prokaryotes, it starts at one point and goes to different points and makes like a complete circle?
Right, so a prokaryotic chromosome is circular. And there's a point at which, I should draw that as a double helix, 2 strands anyway. So there's a point where the replication bubble forms. And this is a spot where the 2 double strands have become denatured. They've melted, they've separated. We've got a double helix, but then there's a small region where, which is called the origin of replication. Try remembering that. Where replication starts. What sort of a base pair composition, what sort of nucleotides, which bases do you think you find in origins of replication? The only thing you need to know here is that what's just happened? You had a double helix, and then part of it did what? Denatured. So which nucleotides are going to make it easiest for bacterial enzymes to melt part of their genome? A's and T's, if it was RNA, or A's and U's. So origins tend to be, in both prokaryotes and eukaryotes, AT rich. It's an easy way for scientists to figure out where origins of replication might be. They look along a chromosome until they find a sequence that's got long sequence, these are not usually short things, that generally have much more A's and T's than the rest of the chromosome. Those tend to be where chromosomes start to replicate. It's where the enzymes have the easiest time pulling the 2 strands apart. And of course, you want to separate the 2 strands because, as we learned from Mendelssohn and Stahl, you need single strands to act as templates for synthesizing semi-conservative replication. So that gets at that second point, origins of replication, number and base pair composition. Just talked about base pair composition. What about number of origins? Bacteria have 1. How many do we have on our chromosomes? 1?
Do we have more than 1?
3?
It would be multiple, depending on which gene you wanted to.
So, quick exercise. Our biggest chromosome, do anybody know which of our chromosomes is the longest chromosome? Chromosome 1, by definition, because we've numbered them based on size. So here's a little exercise for you to do in class. Right now, you can do a partner exercise if you want, share with your neighbor. A few pieces of information first. Here's human chromosome 1. It's 247 million base pairs long. And eukaryotes, our DNA polymerases replicate about 1,000 nucleotides or base pairs per second. So from there to there, all of our polymerases just synthesize 1,000. That's pretty fast if you think about it. Sticking nucleotides together at a fantastic rate. But even that fast, how long would it take a cell to synthesize chromosome 1? That's the question. So work on that for a couple of minutes, and then we're going to come back together and discuss what your answer is.
It was 4 minutes.
4 minutes?
That's 8 hours. Almost 3 days.
Yeah,
Like 68 hours.
Yeah.
We've got maybe I should make this the next Socratic quiz. How long is it? No, we want to know now. Okay. So if you've got 3 billion base pairs, or nucleotides, either one, and synthesis occurs at 100 nucleotides per second, That's how many seconds? That's 30 million, right? Am I good so far? Somebody check my math, right? You get rid of 2 zeros and 2 zeros and you're left with 30 million seconds.
I thought you said 1,000.
Wait, how do you?
Oh, did I say 1,000?
Yeah.
Okay, let's do 1,000. 3 million seconds.
So did I.
Hey,
Okay,
we're using the wrong number.
3 million seconds?
No, we said 247,000.
That's the nucleotides.
Yeah, 3 billion is close enough.
Okay, it's not gonna be one of
No,
the
it's
exact
not.
number.
That's all, then we'd be a different species altogether.
Okay, okay.
68.
Let me ask, okay, let's take a quick poll then. How many of you got answers in terms of it would take a cell hours as opposed to days? Hours as opposed to days.
No, I took a long.
Okay, days as opposed to months. How many people calculated in like 0 to 1 to 28 days, 1 to 31 days? Okay, anybody over 31 days?
No,
Okay, so on the order of a month then, we're agreeing. DNA polymerase is damn slow. Oh, wait, no. Sorry, what's the opposite? Our chromosomes are too damn long. What does this have to do with the origins of replication? This is why our chromosomes have multiple origins of replication. Because it would take one enzyme, like a month, to synthesize our longest chromosome. So you have to have lots of polymerase molecules at a time working to synthesize each of our chromosomes. But maybe I should step back a second. Why is it such a problem that, who cares if it takes polymerase a month to synthesize a chromosome?
Tissue repair.
we would possibly deteriorate.
Basically, we're only tissue repair.
So what has to happen, yeah, tissue repair, what has to happen, what's waiting in biology that we've already talked about in class, what's waiting for DNA synthesis to happen? What can't happen before synthesis is complete? Cell division. Does it take a month for each of your cells to divide?
No.
How long did it take you to become a living, free living organism?
9 months.
9 months. 9 months. If it takes 1 month for polymerase to synthesize one chromosome, there would be 9 cell divisions, 9 months. We'd go from a 1-celled embryo to like a 1,000-celled embryo in 9 months. Does that sound familiar?
Probably not 1,000.
No. We know inherently that our cells divide faster than once a month. We would not be the size we are now if we had to wait a month for our cells to divide. So, yes, eukaryotic chromosomes have lots of replication origins. That means our chromosomes, instead of the example I drew of a bacterial chromosome, our chromosomes have multiple replication origins all at the same time. So here's a linear chromosome with some parts that are melted and DNA polymerase is synthesizing. So that might be an example of a chromosome. Lots of replication origins, lots of replication bubbles. Now I did something here on purpose in that drawing. Does that look, made one bigger? What does that tell us about that replication origin? What does it mean if it's bigger?
It's full of A's and G's.
Let me draw in the fact that there is some synthesis going on. So parts of these replication origins are where replication begins. Right, so here are some of the newly synthesized strands. I'm being very simplistic here. We're going to go into a little bit more detail next. That bigger bubble, how much DNA has been synthesized there? More than the smaller bubbles. This is true. I'm not just cartooning this and making this up. When our chromosomes have multiple replication origins, some of the regions of our chromosomes start replicating earlier than others do. So if you actually looked at a chromosome during synthesis, you'd see something like this. You'd see big replication bubbles, which means they started replicating earlier. DNA synthesis started earlier in time than ones that maybe just started synthesizing DNA, which are going to be smaller if you take a snapshot of a chromosome. They're going to look smaller because they haven't had, the polymerase molecules there haven't had time to synthesize a lot of DNA yet.
The one to the far right?
Yep.
It replicates and replicates and replicates, and then it splits, and then it gets even bigger once it links with the second one.
That's right.
And then they just,
Replication bubbles eventually run into each other because we know that something else that we haven't talked about yet, but synthesis occurs bidirectionally in both directions from a replication origin. So eventually these bubbles grow and grow and grow and they run into each other, and that's how synthesis is accomplished. You synthesize chromosomes not from one end straight through to the other end, but in short little bits. And then we have to start talking about leading strands, lagging strands, and Okazaki fragments. Or we don't have to, I suppose.